Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you to the webinar today on progress on organic naked barley breeding. So today I'm very pleased to introduce Karl Kunze, who is a graduate student in the School of Integrative Plant Science at Cornell University. He works in Mark Sorrell's lab in the plant breeding and genetics section. Today, he will be discussing his work on organic naked barley breeding as part of a NIFA OREI-funded research project on multi-use naked or hull-less barley. So Karl will tell you a little bit more about the project, and I'm going to hand over the screen control to you, Karl.
There you go. Great, thank you. All right, I'll just go through. I'll skip the introduction since it's already done. But my name's Karl. I work with Mark soils and small grains. My research interests include what I'm going to be talking about today, which is some organic naked barley, weed compatibility, and disease resistance. And aside from this project, I also work on our conventional winter malting barley program, particularly looking at dormancy and germination and breeding methods. Some of the larger goals I hope to answer with my research is how can we leverage the technology that we have in breeding for organic research? How can we develop relationships between breeders, farmers, chefs, and consumers so that we can breed crops that benefit the interest of everyone involved? And through this project, I hope to learn some skills over time about how you can develop new crops into new markets. All right, so some of the results I'll be presenting on are not just myself, but work that our whole project has been doing across the country on working on developing multi-use naked barley for organic systems. So in brief, this project is funded by NIFA for 3 years, and it's been currently renewed for another 3 years until 2022 at least. I'd have to go into the details of that. It's longer, but current participating states are Oregon, where the project is led by Bridget Mites, in Wisconsin with Lucia Gutierrez, Minnesota with Kevin Smith, California, and then New York. The main objectives for this project are kind of a lot of applied field and breeding research, barley education, and outreach events. So what is naked barley? Naked barley is controlled by a gene called NUD that determines whether it's naked or covered. Covered or hulled are sometimes used interchangeably. I'm going to try to be as consistent as I can to use covered. It arose as like a kind of a single mutation back a few thousand years after barley is domesticated. The dominant phenotype, so to speak, is the hull is where the hull adheres to the kernel, and when it's recessive, where the NUD allele is absent, the hull falls off the kernels. Some of the traits that are related to naked barley that we've been measuring that are important include ones that threshability, embryo damage and germination, seedling vigor, test weight and yield, disease resistance, and more indirectly beta-glucan content. I will try to be, again, as consistent as I can. I may have made a mistake somewhere, but covered barley is what I'll refer to as hull, seen here at full pint example, and then the free threshing lines will be naked barley. So one of the ways that we've been evaluating how naked barley is doing in these different environments for participating research institutions is that we have these regional trials. And the goals of these are to see what lines work well for potential variety releases, future breeding lines as parents, and to kind of develop experience in growing naked barley in these organic conditions. There's about 20 to 25 lines replicated about 3 times in each of these winter and spring trials, and they're grown at different locations throughout the country. So I'll be presenting the work that we've done so far, which hasn't included California yet. Our renewal project does include California, so they're planning out some trials now. But mainly in New York and Oregon, we grow both winter and spring. In Wisconsin, there's 2 years of spring data and 1 winter data, and then Yeah, in Minnesota, there is only spring. So through this, we hope to learn which lines grow well in which regions and make selections. Another big part of our project are diversity panels, and these are kind of a way of doing like pre-bean selection. These lines have been collected and made into kind of double haploids that are crossed by parents that are well adapted from wild to parents that are kind of like wild relatives. And in this fashion, we're able to try and maintain some of the adapted traits that make barley grow well in the United States while also maintaining some kind of genetic diversity. There are both spring and winter diversity panels. And so the winter diversity panels, of course, can be grown in places like Oregon and New York, where there is still some winter survival issues, but it's a little more difficult to grow in spring in places such as the Midwest, where conditions are more harsh. Some of the notes that we take on these diversity panels and goals that we have for them is to do things such as genome-wide association studies, measure traits that are easier to measure in like single plot rows as opposed to larger plots. And so because of that, we can then score and measure more traits on more lines in less amount of space. So these are kind of some of the traits of interest, where I will briefly go over some of the quality traits, although there will be a greater focus on a separate webinar. Some of the agronomic traits that has been a large focus, including yield and test weight. Something we do, particularly at Cornell, with pre-harvest sprouting. I'll talk about some of the disease traits. Some of them are region specific, some are universal across the country and particular to organic conditions. And then at the end of the webinar, I'm going to be talking a little bit about weed competitive ability and what research I'm working on here at Cornell to find some ways to address how we can measure that better. So quality traits. So one of the principal uses for the multi-use naked barley is we aim to make naked barley usable for malting quality. Barley itself has been the optimum grain for malting and brewing distilling. It's almost like a perfect, nothing's perfect, but a really good package of the right amount of carbohydrates and starches, but also the right enzymes that when you malt the barley, you can turn those sugars and starches into something that the yeast can use when you're brewing. Currently, a high majority of the barley used for malting is with covered barley because up until recently, currently the covered barley in the hull is very useful for filtration in the brewing system. Right now, up until this point, when you use naked barley, it can be a little bit difficult as beta-glucan can get stuck and there's not that hull. Advances in brewing technology, however, are making naked barley a greater viable option that can mitigate these disadvantages of not having the hull, but it still remains to be adopted a little bit. Naked barley also has higher potential for higher levels of malt extract, another trait that's very important in malting quality. And below are some of the traits that we generally select for in the field if we can. And some of these include like large and soft kernels, round and plump grains, low to moderate beta-glucan, and a mid-range protein of 10 to 12%. Another aspect of our multi-use end goals is food quality. Barley is rich traditional culinary significance around the world, but it's become less common. One of the reasons for this is that wheat tends to have somewhat better of a profile, particularly with gluten. But food barley still has a lot of high potential in nutrients such as beta-glucan and protein. So for food quality, we kind of want to have a little bit higher beta-glucan and higher protein. And one unique advantage of naked barley, because it doesn't have the hull, is that it has this whole grain nutrition status. And the third avenue for our multi-use is feed quality. Most global barley production is used for animal feed. Feed barley lines, because they haven't been bred and selected or fine-tuned for these malting quality characteristics, have sometimes a little bit more genetics that can make it more hardy, and don't have to be as picky about the grain quality. And as a result, you can produce really high yields with some of these lines that aren't constrained. One area where naked barley has been selected for before this project was in the 1980s in Canada to feed some of their non-ruminant animals such as pigs. And in fact, some of the lines that we've been testing from their program for our springs have done pretty well for things such as spot blotch resistance. However, most of these will be in a separate webinar, so I'll leave it at that for now. Just a brief overview of the quality traits in the field. Quality traits such as beta-glucan and protein vary greatly by the line you use and by the environment. Since naked lines haven't been traditionally selected for brewing quality, a lot of them are high in beta-glucan. And so we'll, in the future, need to, when we're selecting, select for lines that are a little lower in the beta-glucan. In New York in particular, with what we've seen with our values is beta-glucan can be very high. The numbers here is like 500 parts per million, and the target range is supposed to be less than 200. In spring, our protein values have been very high, sometimes greater than 12% for many of the lines, particularly in drought years, and beta-glucan has also been very high. So for this trait in particular, we need to work on breeding to lower them. The next section we'll be talking about is agronomic traits. So as I mentioned earlier, we have winter and spring barley variety trials. Just briefly, like, what is winter barley? Winter barley is kind of barley that requires like a period of vernalization or a period of cold temperatures before the barley can flower. Associated with this trait also include genes that aid in some degree of winter tolerance. And comparatively to some, in New York, in our experience, compared to other grains such as wheat, barley tends to be a little bit more sensitive than other cereal crops to winter survival problems. And the other type that we're also testing is spring barley. Spring barley is generally grown in like April or whatnot, and there's no vernalization requirement. However, if you were to plant spring barley in the fall, initiation of the flowering would perhaps occur in the winter and eventually kill the plant. So where do we grow winter barley? Currently in the Northeast, the northern range for winter barley is the New York and Pennsylvania area in the Northeast, and most areas in the Pacific Northwest. In the Northeast, we plant generally in mid-September to October. You plant too early, you have issues, potential hedge and fly issues, and heading dates maybe occur too early in May. But if you plant too late, the barley can't establish itself, and then you're going to have a greater chance of winter kill. And in Oregon and Washington, tend to plant winter barley in mid-October. As climate change occurs at a greater rate, this may change, but at the moment, winters in the Midwest, up in New England, such as Maine and Canada, have winter conditions that are too severe for reliable winter barley. Often and whatnot, the temperatures can get very low, particularly more inland you go away from the coast or the Great Lakes, and snow cover can be variable. And so if you have a combination of very low temperatures and low snow cover, that exposes the barley to the elements of nature and can be a problem. So the general life cycle of organic winter barley is you plant in September, to October. The vigor occurs right after planting, and we've kind of measured that with vigor estimates and stand counts. Depending on when temperature is low, barley can be very well established by then. Then, after the dormant period, in New York in particular, from about November to about April, the barley is pretty dormant; it doesn't grow, and this is when we score for winter survival, just to check to see if there was any damage. Once April hits and temperatures get higher, this is when the barley growth starts to increase. And then we can start measuring these traits to see how quickly they're growing and see if they're out competing the weeds. Barley then heads about mid-April in Oregon and late May to early June in New York. Based on some evidence in Pennsylvania and whatnot, if we find that if you have grown barley or planted barley too early in the fall, sometimes heading can occur too early and cause some frost damage. And then as the season progresses for maturity and harvest, in New York, we harvest barley in early July. In Oregon, it's in late June, early July. And in Wisconsin, where we were able to have a successful winter barley field trial, they harvested in about mid-July. Spring barley. Spring barley is probably the predominant type of barley grown in most regions in the United States, and mostly grown in the Midwest, up in New England, Canada, and Canada grain regions. And a lot of the model species that we use for genetic studies and whatnot are mostly spring barley. Currently in these regions in the Midwest and up in New England, the winters, as I mentioned earlier, are too severe for winter barley to be grown. Climate change may change this, but at the current moment, spring barley is the way to go. And spring barley, as you get further and further west towards Dakota, is really favorable because at the time you grow it to the time it matures, there's not as much rain to worry about when the barley nears maturity. And so this is really beneficial for seed-borne diseases, and you also don't have to worry about the potential of rain resulting in pre-harvest spreading. And based on the data I've seen from the Pacific Northwest and milder Northeast regions, spring barley can be a little bit challenging. To the left is a picture of a really good spring barley year we had in New York, but midsummer and humidity can cause unpredictable rain events. And when planted kind of in April, when everything's also getting going, barley doesn't have as much of a head start and is competing more with the weeds for light resources. So the kind of life cycle of growing spring barley, it's late March and early April for most regions, maybe later in New York or in the Midwest if temperatures are pretty low. Barley tillering vegetation growth starts approximately 2 to 3 weeks after planting. Heading occurs in late June and July in most regions. And for maturity and harvesting, New York is late July, and Midwest and Oregon is about early to mid-August. So I've been already going over this a little bit, so I'll go quickly over this, but what is winter survival? Its ability to survive winter conditions. We score this by looking like about now how the estimates of daily temperatures are consistently above freezing. What factors do we think affect winter survival? Some of them include like degree of establishment before freezing temperature, the severity of the winter, snow cover, and field effects. The challenges with winter survival include it's a very difficult trait to quantify accurately due to numerous environmental factors. Winter survival pressure is variable as well between years. So in a reliable year for the pressure, we can only detect it every 2 to 3 years in New York when we have appropriate conditions. And there can be times where we can have 3 years of winter survival where a line does really well, but then the 4th year, it can be detrimental. So for example, we have this line called 10-1150, name doesn't matter, that's had pretty good winter survival for the last 3 years. However, this year in 2021, even in a mild year, the same line that we tested in this field was the winter survival was only like 10%. And there's many factors that we don't know that could have contributed to that. So it's important for lines to test for winter survival that you do it over many years. Let's get that because I went over too much. Another trait that we are interested in with looking at barley is height and lodging. Height is an important characteristic for barley, particularly for organic barley. Tall barley can be really useful for wheat competitive ability, but you got to be careful because if you have too tall of barley to select for, it increases your susceptibility to lodging. Too short of barley, not always, but can be less vigorous, and that's a problem in organic conditions. You want a barley that's very vigorous early on to help grow up against the weeds. And a related trait to height is lodging. This is the capacity in which barley simply falls over for many reasons. Some of these factors that contribute to lodging include height, the weak stem straight, over-fertilization, soil structure, and there's just also a lot that's unknown about it. Many breeders have tried to quantify lodging and to some degree is successful, but there's always another factor that makes it more complicated. And of course, things beyond the plant's control that include weather events. So to the left is one line that we've had that is highly susceptible to lodging, as you can see there. Generally, results for our lodging is that heights are taller for winter lines compared to the spring, 70, 90 centimeters in winter and 50 to 70 in springs. And for lodging, it's a relatively minor issue for the winter lines that we've tested in New York. Some spring lines are very susceptible. Another trait that we test, particularly in New York, and put a lot of focus on is this trait called pre-harvest sprouting. It's when the barley grain starts germinating in the field before it's harvested. It's a particular problem in New York, where rain events can often occur during our harvesting windows. Most of the time we get very lucky, but in 2018, we had this very exact scenario for the reason that we breed for pre-harvest sprouting. Selection for high germination, particularly in some spring barley germplasm historically, were places where you didn't have to worry about it, has inadvertently resulted in lines even being that much more susceptible. And so as a result, when we're testing our germplasm, we want to make sure that we have lines that are adapted here that should these rain events occur, they won't germinate in the field. So in our program, we test most of our trials for this trait. Just briefly going over it, what we do is that we harvest spikes at physiological maturity. We dry the samples for about 3 to 4 days. We put them in mist chambers, and then after about 3 days of this simulated rain event, we test them for each entry. And then the scores are based on how much the seed grows in those mist chambers. So for the results for these, what we have found with our winter variety trials is that we haven't had an issue at all. Part of this is due to the fact that in June and early July, when we harvest winter barley, we haven't had a problem with rain events, and that most of the lines are genetically resistant to pre-harvest sprouting. There were a couple lines which we already knew ahead of time that would be very susceptible to pre-harvest sprouting, and they were. So they were pretty good controls. For our spring variety trials, we unfortunately have had some inconsistent data due to stressed environments that our spring barleys were subject to, which made for some uneven data. But overall, more of the lines did have some increased pre-harvest sprouting susceptibility. Of course, one of the biggest traits that when breeding most crops that you're after, of course, is kind of the yield. And in our program testing for yield, we have found that depending on the environment and location, yields of some naked barley lines are comparable to conventional ones, particularly here in New York. One big factor that's very important for this is having really well managed organic land. In addition, the cooler temperatures in April and May, when winter barley gets a head start, is very ideal for its development, as barley is a kind of a cooler temperature cereal crop. And so it really appreciates the cooler temperatures that exist here. Another interesting finding that when we planted these in our organic conditions is that the weed management that we needed for winter barley was very minimal. In my 3 years of working with both diversity panels and our trials, I really didn't have to do any kind of weeding whatsoever. Weed pressure was really minimal, and one of the factors that may have helped this was just maintaining the soil structure and not doing much tilling in the early months. However, yields are still lower than conventional barleys. Some of that is due to the fact that conventional barleys have been bred more for these conditions, and so more breeding needs to be done. But also the lack of hull does reduce the absolute yield that you measure when taking it out of the field. So that's something to consider. Overall, as for places in Oregon and Wisconsin, I can look at the data a little bit, but in terms of some of the practical field applications, I don't always know since I'm not there. But some of the yields in Oregon and Wisconsin were definitely lower than their conventional counterparts. So in the future, now that we have had some data about testing these lines, we will need to develop breeding lines from parents that have shown like high performance in target environments. As climate change affects the growing conditions across the U.S., keeping these lines and keeping a germplasm open for developing winter barley and testing them in these new environments may become really beneficial in the long run. As for spring barley yields, outside of Oregon, many of the yields have been kind of really low. They haven't been great. Some of the reasons for this is perhaps some of the germplasm is adapted to more northwest conditions. And probably one of the large factors that in some of these environments, especially if barley is planted late, as it can often happen, barley does not like high temperatures during development. I can't speak for, of course, the Midwest and the Pacific Northwest as much, but New York, many of the times when the barley is developing in May and June, we're having temperatures that are really high and humid. And barley doesn't always appreciate that as much. The other factor that makes organic grains very difficult is that the weed pressure is really high. Because when you're tilling the soil up and planting the barley in April, or in our case, is unfortunately we've had to plant as late as May. That puts the weeds at almost a similar rate in competition for the light and resources as barley. And so as a result, we've had to do a lot more weeding management. The other big takeaway in New York, which is absolutely essential, you need well organic managed land. At one of our locations in our research farm at Freeville, the land there is very well managed, and as a result, even with drought conditions that we've had, yields have done decently well. But at other locations where we've tested where organic is field land management just becoming a thing and it's not fully developed yet, our yields were significantly lower. So you really need good land for organic spring barley. Another agronomic factor that we're interested in is threshability, and that's the capacity for which the hull completely dissociates from the grain. We have found in our trials that a number of naked barley lines, the hull doesn't always want to fall off, which may be a challenge to the notion that it's just a single gene, but it's also a very highly environmental factor. And to put it in another way, some lines are a little modest, so to speak. We've had to pay much closer attention to score our lines, both in the variety trial and diversity panel. And so with the diversity panel in particular, we're looking into scoring this and seeing if we can find a genetic region that's associated with why some of the hulls don't always fall off. For specific types of grain for the winter grains in New York, we haven't seen as much of this being a problem. The hulls still adhere, but not too bad. But in particularly with the spring grains, we've had a lot of lines where the hull doesn't completely fall off, and you need more processing to remove the hull. And the big takeaway here is that the naked phenotype can be complex in different environments. Storage, one thing that's been very humbling here is that when it comes to the post-processing of breeding lines and whatnot, is that every state has different stages in storage and upscaling of varieties. So once again, I can only speak for New York. We do have an established foundation grow-out system, but we only do for conventional. And as a result, when we've had to store grain, such as for like our buck barley grow-outs, whatnot, it's always been a challenge as a breeding program to kind of sometimes store the grain in certain locations. And in Oregon, large amounts of grains. So one area of improvement when thinking about naked barley and value-added chain development and whatnot is that we got to think about more about when we take the grain off the field and when it becomes too large for one breeding program to handle is where does the grain go and how can we make sure that it's more streamlined so it gets to the markets that we need to go. Diseases. So going on to barley diseases, one of the other big factors that affect barley in these field conditions are barley diseases, and these can become a little more exacerbated in organic conditions. One of the biggest diseases we have to worry about in barley, both conventional and organic, is Fusarium head blight. Fusarium head blight is a serious disease for both the barley and wheat in the Northeast and Midwest. Fusarium's alternate host is corn stubble, which, because corn is so ubiquitous in our agricultural systems, it results in a lot of excess inoculum every year. This disease takes advantage of kind of the spaces between the kernels and these kind of microclimate environments where it can grow, which can be an issue for some 6-row barley lines. And the big difficulty with FHB is not exactly the disease itself, but the byproduct that it grows. And this is called deoxysilanol, or DON for short. And it's a vomitoxin that is not something that we really want in our foods. In conventional systems, we've both relied on genetic resistance, but also to use pesticides to reduce the Fusarium head blight incidence at flowering. In New York, we have a separate nursery dedicated line to just testing Fusarium and DON. What we do is that we inoculate this nursery with the Fusarium head blight at flowering, and we score it kind of 3 weeks later. With our winter naked barley variety trials, we've done this with the winter barley for 2 years and the spring barley for 1 year. Overall, of course, because we inoculate, we expected the FHB severity incidence and DON values to be really high because we're intentionally putting that pressure on it. However, given the values, we have found that DON values are really high. And so one of the major takeaways from this is that we're going to have to really rely on breeding for these lines in order to reduce the DON values in organic barleys, especially because we don't have the advantage of using pesticides. Another disease that is particular to organic grains that we haven't had to deal with in conventional grains as much, both wheat and barley, is covered in loose smut and the seed-borne fungus in the grain. Essentially, if you do see smut in your grains and you see these heads as shown here, it's likely that your seed lot's most likely contaminated. Historically, seed treatments have made smut kind of a non-issue in conventional barley, but because in organic we don't have this advantage of pesticides, we'll also have to rely more on genetic resistance in the barley itself to be resistant to this fungus. Fortunately, genetic diversity does exist, but we're going to have to work a lot more on selecting for this. Another barley diseases that are important are rusts. Depending on where, this is where it becomes a little more region specific. In the Pacific Northwest, there's stripe rust. It develops in the cool, wet climates during the April months and then becomes a problem once the barley starts emerging in April. A resistance for these, of course, is achieved by fungicides and selecting resistance varieties. Once you get more towards the Midwest and the Northeast, leaf rust becomes an issue. In New York in particular, we've only had 1 year where leaf rust has been a significant issue, but I understand that leaf rust can be more of an issue in more hotter climates in the Midwest. And another disease that we've been focusing a lot in New York or trying to figure out is scald. Scald is more particular to winter barley as it prefers cool temperatures during the barley vegetation growth. Symptoms occur both in New York and Oregon, but it's been a particular problem for us in New York. Selection for resistance has been a little difficult as our plant pathologists that we've been working with have had difficulty in isolating this fungus. And one of the reasons you want to isolate this fungus is so you can take it independently and then maybe inoculate barley plants in the greenhouse and kind of select for the resistant lines. Or better yet, develop a marker that can help you select these lines. However, in our experience so far with scald in both conventional and organic barley, we have found that the symptoms of scald are not only hard to replicate by inoculation, but within the field they can be very field location environment specific. And so we'll need to do a lot more research to understand the mechanisms of scald. And of course, variety resistance is also going to be important in organic systems since we don't have fungicides as an option as much. Another disease that we're concerned about is spot blotch. Spot blotch is a barley fungus that kind of overwinters in barley stubble and is also seed-borne. It really likes the warm, humid climates, and this is particularly problematic in our spring barley. Spot blotch symptoms appear after barley flowers, and the symptoms can be often accelerated by rain events. Most of the lines that we've tested, in New York at least, have been pretty susceptible, but to varying degrees. Some of the CDC lines from Canada have some resistance, and there is some limited variation in susceptibility and diversity panel. Big takeaways from spot blotch resistance is that timing of scoring is important. And one of the biggest solutions, of course, is that we need to continue to test more lines to select for resistance. Overall, for disease resistance in organic grains, cereal diseases are going to be most likely the biggest bottleneck in organic grain development. Selection for resistance in winter and spring organic barley for the respective pathogens will need to rely on techniques just beyond field phenotypic evaluation if we hope to have some faster progress. And these can be, of course, achieved by processes such as marker-assisted selection, and of course, putting more research into these pathogen isolation and developing the markers in the first place. The other factor, which is hard to control in breeding systems, since we're limited on what fields we can do select our trials on, which is cultural practices that can play a large role. And these can be things such as crop rotation, how the soil is managed, and where and when you plant. So big takeaways for winter barley management, as mentioned earlier, is if you plant it early enough and the winter is not too extreme, winter barley performs really well in organic conditions. Weed pressures on our farms have been very minimal. In our 3 years of growing organic and winter barley, we haven't had problems with the weeds at all. One of the biggest downsides to organic winter barley is that a lot of our lines are very susceptible to the Fusarium head blight. And so we're going to have to really work on developing disease resistance to that. That, and of course, yield still needs to be improved. Spring barley takeaways in New York, and to a lesser extent in Minnesota, Wisconsin. We've had some, in our 3 years of growing some of the spring barley, we've had very challenging years for spring barley development. Due to rainy Aprils, we've had some late planting dates, as late May, and sometimes some drought-like conditions during the vegetative growth, and some land that hasn't been the greatest at our secondary trial locations. As a result, the yields in 2 of our years were really low. We needed really persistent weed management, and of course, the barley diseases were a constant threat. The big factor for New York is that we're going to need much more breeding and evaluation to find lines that are a little more adapted to the New York growing environment conditions, because many of the lines, even the diversity panel, didn't always perform that well. So after listening to this, there's many places where you can purchase naked barley, where you can purchase winter naked barley. Some of the lines that we've grown that are available for purchase is Buck, and you can get that from the Washington State Crop Improvement. Another line is Streaker, which is a blend that contains colored barley. You can get that at Hummingbird Wholesale or Territorial in Oregon. And some other lines in the Northeast include a Maize 10 from Virginia State. For spring naked barley, if you're interested in growing those, there's some lines from the Canadian, from CDC Clear, CDC McGuire, and CDC Ascent. Those are from Canada. Meg Songs and Havener, Washington State Crop Improvement. And so for some of our colored naked barleys, Purple Valley and Kalma and Carmen at 202 Grains in Oregon. One of the projects that we've also been working on, in addition to variety trials, is in New York. We did start our own breeding population. What we're trying out is crossing the top performing naked barley lines that we evaluated before this project began to some of the covered barley lines that we know grow well. And we hope that from these lines that we're going to be able to find some lines that do really well in both, that have really good yield from the covered barley germplasm, but also the desired naked traits such as the naked trait, and have lines that do well in organic conditions. So how much time I have here? I'll have to be very quick. So one of the bigger traits that's specific to organic breeding is weed competitivity. Weed competitivity is the ability for the crop to compete or tolerate the weed pressure that exists in the field. This is an obvious problem in organic systems versus conventional barley, as we don't have pesticides to simply get rid of the weeds. However, due to the dynamics of how weed populations grow, it's really difficult to measure them. Some ways that you can do it include looking at species distribution or manage it as crop rotation. And for barley, some of the ways you can measure this is through vigor, the amount of vegetation growth, and stand counts. So in our project, how do we measure weed competitivity? Throughout in vitro locations, we've been measuring different components of barley vigor as well as weed estimates. We've done aspects such as barley weed ground cover estimates at multiple time points, done vigor ratings, and early height. Looking through these results and trying to get this together, it's been a very big challenge to model accurately due to the environmentally specific nature of weeds in barley fields and weeds in specific environments and specific fields. One possible solution has been to perhaps maybe do some kind of weed seeding, but even consistent weed growth doesn't always cooperate, even when you want the weeds to grow there. So one of the projects I've been working on is to utilize aerial imaging. And so what you do in aerial imaging is using like a UAV or a drone. Using a UAV or a drone to be a potentially useful tool for modeling plant growth in the field. And what you can do with a drone is you can attach a camera to it, have a set flight path, and you can fly the drone over the field, and you can capture like light data, reflectance data, or essentially be taking pictures that you can use and quantify to measure the health of the barley plots and the amount of vegetation growth. The aim here is if we can measure the vegetation growth repeatedly over the life cycle of the barley, we may be able to select for the barley lines that develop vegetative growth at a quicker rate. And as a result, faster vegetation growth and earlier in the season could be a useful method for the barley to outcompete the beets. So the basic process for aerial imaging is essentially you need to develop a flight path for your fields. On target days that you want to measure, you fly the unmanned aerial vehicle with the camera attached using a flight path that you made. You then take those images and you put them through a processing software to put the images together. And then once the images are collaborated together in a process called stitching, you use these stitch images and use another program to assign polygons that represent where each of the plots are, as you can see up on the left here. And once you assign these plots, the software processes for a while on this pipeline called ImageBreed. You can extract these traits called pixel values. So each pixel represents like a light part of the plot. You can model these over time to calculate kind of indexes that are related to the vegetation of the barley. And so the images are a little distorted here, but you can see a little time lapse the same plots over 3 different points. And then you can capture these different rates in growth of more vegetation for each plot and try and model that to see what barley grows the fastest. To the right here is a picture of one of the images, stitched images from this process. So this is just the values over time on the first year that we've been doing this. As you can see, like NDVI value, a higher value corresponds to a higher amount of vegetation. And so as you can see, and the days below, the days after planting. As you can see over time, the vegetation increases substantially and then tapers off towards the end as it reaches maturity. Here in Freeville, of course, this was an ideal location. The weather was good, the soil did pretty well, and as a result, you had a very consistent growth rate over time. However, in places such as Caldwell, on this other field location where conditions weren't as ideal, the growth can be a little more scattered. So what are the applications in organic plant breeding with this? If being able to deploy this, you can provide like a really highly quantitative method of classifying plant growth. It's higher precision than just doing visual estimates. You can even have greater applicability to kind of crops where the biomass is the end product, such as alfalfa or even cover crops. However, there's a lot more work that is needed to move aerial imaging to just split our application to organic systems. There's a lot of things such as learning to set up flight paths and understand the software, finding affordable cameras and drones and software. But over the horizon in the next few years, it could be a really useful tool in organic systems to kind of quantify these vegetative growth rates. And the next steps in the project are for the next part is we're going to be developing a kind of breeding panel called a Nested Association Mapping or a NAM panel for marker assisted selection. And what this is that we take 3 parent lines that we selected based on performance in this project that are common in all the crosses. And then we take 5 lines that through this project, we found performed really well, and we cross them to those 3 parents. And our goal is to develop a really large population, use marker-assisted selection at the early stages of breeding development, and then send partially some of these lines to each program for field selection to eventually, at the end product, select a line that is adapted to each of the respective locations. If you have any questions, feel free to email me. at khk44.edu. And thank you for your time and listening. And if you have any questions, I'd love to hear them.
Okay, thank you, Karl. We're going to start our question and answer period. So if you have a question, you can just type it into the Q&A box on your screen, and we'll be reading the questions out loud until we run out of time. So I'm going to just take a look at the Q&A box and the chat box and see if we have any questions. And I was interested in what you were talking about, while you were talking about the aerial imaging system that you had there, how you tell the difference between the weeds and the barley. Are there differences in the color gradations or something like that? Is there some way to get around that?
Unfortunately not. I should not have left that part out. I don't know why I did. Unfortunately, because of that, we had to kind of time it to the point where to manage it properly, we would fly the drone after we did our weed management. So we would unfortunately have to get rid of the weeds. We couldn't just leave them there. Now, technology is increasing to the point where, and maybe in the next few years, it'll be precise enough where perhaps you can, based on leaf form and function, leaf form, that you can tell the difference between the weeds and the barley. But if your primary weeds are grasses, telling the difference between that and the barley plant is really difficult. And we don't currently know of any way where the emission, the light reflectance of the weeds is different from the barley. So without that differentiation, we kind of had to fly these aerial imaging after weeding. So that was kind of the unfortunate part.
It's cool though. It sounds like it has potential. So
yeah.
hopefully the technology will improve. Okay, so we do have some questions coming in. How would you describe the evolving market for food barley?
Yeah, that's a good question. So the evolving market for food barley it's increasing, but it's probably not as increasing as much. It's not increasing as much as, say, your other grains such as wheat or your emmer or your einkorns. But it's getting there over time. However, yeah, the market still is a little small, but it's increasing.
Is it likely that the Northwest will have the edge in barley quality?
I would say a little bit in terms of disease resistance and whatnot. Yes, particularly if in the Pacific Northwest, when barley reaches maturity, it has a higher or has a drier climate, so you don't have as much of these diseases. In the Midwest, I can't speak as much, but in New York, we definitely have more rain, but there's definitely a capacity for winter barley to be pretty competitive. But yes, I would say the Pacific Northwest is probably the largest advantage.
Okay, thanks. Okay, so here's someone who's interested in soft barley. How soft have you found it? And is it similar to soft wheat? And how do you select for soft versus hard kernels? We had a comment about the hardness from Bridget. Thank you for adding that, Bridget. Bridget is running the project with Pat Hayes here in Oregon. And she said that hardness in barley is measured through a single kernel characterization system, which crushes the grain and measures resistance. We have seen a huge range of hardness in the diversity panel of barley that Karl mentioned. And compared to wheat, these range from the softest of soft wheats to the hardness of durum wheats. So thank you for that, Bridget.
Yes, thank you.
Yeah. Is pre-harvest sprouting related to the longer-term dormancy, a maltster's desire for grain storage?
Yes. Okay. So we've been working on this quite a bit. Our spring barley, conventional spring barley and winter barley. So in our understanding, it depends on what you want for grain storage. So I guess my question is, with this question is, do maltsters in the specific context want longer grain storage or shorter grain storage? Because in the context we've been studying our relation to pre-harvest sprouting and dormancy, we have assumed that we want low pre-harvest sprouting, but still shorter storage, or still like quick dormancy. So we're trying to find the balance between the two. But I would say that if you hit in some lines where it's not selected for, if you have very low pre-harvest sprouting, doesn't that could mean that it could have low dormancy, but that's not always the case.
Are you selecting for baking quality in naked barley?
Yes, yes, we are. But yes, uh, in New York, we don't have the capacity, and so what we have been doing is once we harvest our variety trials, we send them to Oregon, and they've been working on baking quality.
Let's see. Will continued exposure to FHB naturally breed resistance if seed is saved year after year?
We do not have any evidence for that.
Okay.
We have not had evidence for that, unfortunately. So no, we're going to have to. In our experience working with Fusarium head blight or nurseries, we have to repeatedly measure over a number of years the same lines and multiple replications to see what does well, and then we'll use those lines in future considerations for parent lines.
Okay, great. We have a couple more questions. Will the attachment of the hull, the lemma and palea, to the rachis in two-road barley be measured?
Yes, we have been measuring that, and especially in the last 2 years when we've done variety trials and we harvested and realized, oh, the hull's still on the grain. This shouldn't be happening. So yeah, we have a rating scale of about 0 to 4 that we've been doing on both the diversity panel and the variety trials to measure this. And so that's something one of the traits that we've been looking for and selecting against.
Okay, is there data on food nutrition of naked barley versus pearled barley?
Versus pearled barley, I'm not sure. That is a very good question. I'm not sure.
Okay, anyone else, feel free to chime in here. Let's see. Okay, so here's someone who just planted 4 varieties of naked spring barley interplanted with Dutch white clover. So he's wondering if you're experimenting with interplanting to control weeds. And then another comment here, definitely they have a problem with FHB in spring barley in Michigan. So they're wondering if winter barley might mature earlier in Southwest Michigan and avoid that pressure somewhat. So we have 2 different questions here. The first one was about whether you are experimenting with interplanting to control weeds.
Right now, we're currently not. I would like to. I would really like to do all these things, but at a breeding program and given our research farm, we're limited in the capacity in which we can do these. So right now, we're not. But that would be something we've done a little bit before, but that would be something I would like to do. But yeah, unfortunately, with some of these trials, yeah, we only have so much capacity to do things, and yeah, I would like to do that.
Okay. Yeah, and then the second question was about whether winter barley might mature earlier here in Southwest Michigan to avoid some of the problems with FHB.
So we have found that with our winter barley Fusarium head blight testing, we have separate nurseries for winter and spring. We've actually found more variation and better established variation in our winter lines where we can measure it better. So I would say that I don't think you're unfortunately going to be able to escape it if it's winter or spring. but yes, there's going to be less humid conditions in Michigan, I would assume, with winter barley. But speaking to Michigan in particular, I'm not sure how mild winters are and how much snow cover you get. So I'm not sure how well winter barley will survive. But
Okay, we have another comment here from Bridget in the chat related to the nutrition of naked barley versus pearl barley. And so she wrote that pearl barley, where the hull and some of the bran and the germ are physically abraded off, has lower levels of minerals and micronutrients than naked barley. And the reduction will depend on the rate of pearling. However, beta-glucan, the soluble dietary fiber in barley, is located throughout the cell walls of the endosperm, and that is not reduced in pearl barley. What do you consider being the main parameter for baking quality? In my organic naked barley program, I select for content of D. hordenes. Do you agree? Okay, it sounds like
I,
it sounds like that person should be in touch with Bridget. So,
yeah,
you
I
can.
yeah, the reason that yeah, at Cornell we've been a lot more focused on for the agronomic side of things. Uh, I really would like to do the baking side, but at the moment we don't have the capacity to do this yet. But once we can, that would be
Yeah,
great.
this is a multi-state project, and Bridget said again that baking and food quality will be covered in a future webinar, and there is a grad student whom I just ran into this morning at OSU working on this research. So yeah, feel free to look up the Organic Naked Barley project at the link on your screen and contact any of the researchers involved, because I'm sure they would be interested in hearing what you're doing if you're also researching naked barley in your program. So that's one of the great things about these webinars is sometimes they end up resulting in new collaborations because people from all over the world can tune in. So I'm just gonna, okay, I just found one more question here. Oh, okay, yeah. Bridget also mentioned, by the way, that we have an Instagram for the whole project, which is just at Naked Barley.
There's another question here I
Yeah.
see.
Mm-hmm.
It's regarding loose smut.
Yeah,
Andy,
let me ask the question
yeah,
here just so
yeah,
everybody can see, because
yeah.
I'm not sure if everyone can see it. So when you select for genetic resistance to loose smut, do you go for single gene resistance or the tendency to open flowering?
We're not quite there yet. So Bridget is currently working with our diversity panel and developing inoculums for smut, and we're trying to figure out what genetic areas of interest exist in our diversity panel for smut resistance. So, well, we don't know yet.
Okay, great.
Yeah.
Yeah, thank you. I think we've reached the end of the questions here. We're almost out of time, but if you have a last burning question, you can, I'll just give a few more seconds and you can feel free to enter it in. And while we're waiting, I just want to remind you again that the slide handout will be archived along with the webinar on the eOrganic website, and the webinar can also easily be found on the eOrganic YouTube channel in case you'd like to look at Karl's slides again. So, okay, I don't see any more questions, but I'd like to thank everyone who entered questions and also thank you, Karl, for this excellent presentation. And we look forward to more webinars from the Naked Barley Project in the coming year.